Can somebody confirm you can hear me?
Yes.
Thank you. Oh gosh, that's awful. I guess I should discuss the infectious disease project first. Or maybe that finished. I don't know. Right, let's begin. So today is April 15th, and we are going to be discussing and beginning the infectious disease project, as well as Lab 02 aseptic transfers. Sorry, I'm taking some medication here, and here I've got to drink all this. I don't know if you can see that. There we go. Uh, in the next hour, and it really tastes awful. It will probably make me sick too because I'm not doing very well with this medication. But you should also read the textbook through the end of the section on compound light microscopy. This is in chapter 3. You need to do that before we begin labs 3 and 4. We'll begin lab 3 on Thursday. Any question about any of this? All right, let's begin discussing the infectious disease project. And let me state that some of you are not attending the lab. Obviously, it's not any of you guys, but you will be confused. And it's quite obvious to me who's not attending the lab and who is. I can actually look it up because I do get that information. But on your answers uh, to like the worksheet. So if you really want a good grade in this class, you need to attend the labs. Let me shut this down. And my computer is now working a little faster because the Zoom session is it's stopped making the movie to the first Zoom session today. All right, let's go back to here. Talking about the infectious disease project. The infectious disease project is a research project where you will do research on and become an expert at one infectious disease. To begin the project, you first have to pick an infectious disease and then send it to me for clarification and okay, meaning I have to give you permission to do that project. Each student will have a different infectious disease. So the first student who asks for an infectious disease will get it. The second student will have to choose something else. So it come on a first come, first choose, whatever the saying is, first choose, first get. It has to be a specific topic too. Like some infectious disease, I narrow them down. Like some people ask to do pneumonia. And I'll tell you, no, you can't do pneumonia because there's too many species that can cause pneumonia. You have to pick one species that causes pneumonia. Like you can cause Streptococcus pneumoniae, which causes pneumonia. Or you could cause Klebsiella pneumoniae, which causes pneumonia, meaning you have to write it on one species that causes a specific disease. And then there's other diseases which I tend to separate up because they're very popular and a lot of students like to do them, like HIV. I can split it into HIV-1 and HIV-2. If two students ask to do HIV, I'll tell them, no, you have to do it on HIV-1 and HIV-2. They are slightly different diseases, by the way. And the same thing with malaria. You can write it on malaria caused by one plasmodium species as opposed to malaria caused by, I think there's different 5 plasmodium species that can cause malaria. And they're slightly different diseases. Any question about that? So you have to pick a disease and then send a request to do that disease in an email. And then wait until you hear back from me whether you get that disease or not, or if you need to narrow it down or something like that, or if it's even allowed. And if you're late on asking for a disease, you should probably request more than one disease to do. Meaning, obviously, if you're doing it in this week, you can just request one disease. But if you do it, ask for it next week, you should give 2 or 3 different diseases because the first disease you request may be taken by another student. Any question about any of that? So you
Should
become,
we email you through Canvas?
I'm sorry?
Should we email you through Canvas?
Oh, yeah, always email me at Canvas. If you want a quick response, you have to email me at Canvas because my clark.edu email, I will only check it maybe twice a week. Okay, I won't check it any more than that. But the Canvas email, I'll check at least every other day, if not every day. All right, so you can become a specific expert on one specific infectious disease, determining the underlying cause of the disease, what microbe does to the human body, how we treat the disease, how the social, economic, and political factors affect our ability to deal with the disease. Sorry, my medication is causing a lot of phlegm in my throat, and I'm having a difficult time talking. And God, that medication tastes terrible. I also have to drink a lot of fluids, so I got something I like. As you become an expert on one particular disease, you can read through the rationale, the project format, pick an infectious disease, and check with your instructor about your disease. If no one has requested your disease and your instructor agrees with it, you'll get it. But you first have to email it to me to see if anyone else has a disease and if I want to narrow it. And then learn about your assigned disease. You'll need to do research. And then you're going to write a written report, submit a written report. It must be typed, single spaced with 1-inch margins, and also it must have 12-point font. If you notice, I've got reading glasses. Anything less than 12-point font, it'll be difficult for me to read. And I should mention that some of you are putting your answers to the lab questions in less than 12-point font. Please increase your lab point font size to at least 12-point font. The document must have cover information with your name, the date of the class, the name of the disease, the date, but that should not be a cover page. It should be simply the information above your first page of text. Do not put it on a cover page. Put it on your first text page. Any question about that? Then you'll have the text, the main body of information. The main body should not exceed 5 single-sided pages. Anything less than 4, you'll get a penalty. Anything more than 6, you'll also get a penalty. Now, this is the text information. It does not include the references, and it does not include any pictures that you submit. Just the text, you're right. The text should describe all aspects of the disease in the report content, which I'll talk about shortly. The text will be worth 60 points, an additional 15 points for grammar, spelling, flow, and readability. Any question about any of that? So the text will actually be worth a total of 75 points, including grammar, spelling, flow, and readability. The text should be written in your own words wherever possible. Where not possible or where you want to quote someone, put it in direct quotes when absolutely necessary. And then you have to state where you got that information from. Any information that is not common knowledge, whether it is directly quoted or paraphrased, meaning written in your own words, must be credited with the proper source within the body of the text immediately after you give the information, not at the end of the paragraph where you list a whole bunch of sources. By using an in-text citation. If you do not do that, you will be plagiarizing information. Like, let's say you state the number of cases of COVID-19 in the United States. I'm sure it's over 1 million now. I don't know what the number is. It's not common knowledge. So you must, you don't have to put it in quotes, but you must put it in an in-text citation where you got that information. Now, when you're giving something that's common knowledge, like you state that colds and flus are more common around the winter time, that's common knowledge. You don't have to put in-text information there. Any question about any of that?
I have a quick question. If
Yeah.
it's a few sentences long, do you want us to make sure to say like the last three sentences was used for this citation if it's the same citation? Does that make sense?
There is an exception, like let's say you have a paragraph and you're always citing the same source for each, let's say, fact that isn't common knowledge. Like you have one fact in sentence one, a second fact in sentence two, a third fact in sentence three, a fourth fact in sentence four, and they're all the same source, in which case you don't have to give the in-text citation after each sentence. You can put it after the end of the paragraph. That is an exception. But if sentence one has one source, sentence two has a different source, and then sentence three, four, and five each have the same source as sentence one, then you have to do it by sentences, meaning you have to do it 5 different times, with the second one being different, and then the first, the third, the fourth, and whatever being the same, because they're not all the same in the paragraph. But if they all are the same source in the same paragraph, then you can put the in-text citation at the end of the paragraph. Okay? And that's the only time you can do it at the end of the paragraph, when they all in the paragraph are from the same citation. Otherwise, they have to be after each sentence when you give a fact or you quote somebody that is not common knowledge, or when you quote somebody at all, you have to give an in-text citation. Okay? So any information
Perfect,
that is
thank you.
not common knowledge, you have to give an in-text citation. Otherwise, you'll get docked on the in-text citations. There's going to be a question? No question? All right, let me show you what an in-text citation looks like, because students frequently get that wrong and then they get docked because they don't have the correct in-text citation. Let's come to my syllabus. No, let's come to the lab manual. That way, if I change it, no problem. All right here. This is problematic when working with microbiology lab because we generally want to work with a pure culture. Don't need an index citation there. That's common knowledge. Cultures that contain one or more species of microorganisms. In the mid-1800s, Louis Pasteur developed a technique of making aseptic transfers to minimize the risk of culture contamination. Okay? The accidental inoculation of unwanted microorganisms involved in our peer cultures and sterile media. So that right there is not common knowledge. So it should have an in-text citation, and we're just going to say Pasteur. I have no idea when this was, but we'll just say 1880. So for the in-text citation, you have to give the author's last name, assuming he's the only author, and then the date that he published it at. Okay? Any questions about that? That's the in-text citation. If there's one author, if there's more than one author, like there's two, then instead of going Louis Pasteur, let's go ahead and just make this a break here, which I guess I should have that there. We'll do something. I have no idea who worked with Pasteur, so let's go Koch. And oh gosh, what was the name of that guy? Oh, I don't remember. We'll just make up somebody. So there's two. We'll go 1882 here. And that should be Anne, sorry. So if there's two people who publish together, you give both last names, Koch and Pasteur, 1882. If, on the other hand, you're quoting for more than two, you give the first last name's author, and then you go et al. And then we'll go 1883, just to make it different. So that would be something like Koch, Pasteur, Amin, Bishop, something like that. You can go et al. So in the in-text citation, you'll frequently see Pasteur, 1880, or Koch and Pasteur, 1882, or Koch et al. If there's more than two, 1883. This is how you do the in-text citation correctly. Any question about any of that? Now, let me back all of these out. Boy, that's going to be a long time. There we go. And actually, I just don't need to save it, but I'll save it here. Any questions about in-text citations? They are worth 10 points. You may use any commonly used citation format, APA, MLA, etc., but be consistent. If you use one format, use the same format throughout the assignment. Your sources you cite in your paper should be scientifically appropriate and reputable. So U.S. government information, USDA, FDA, CDC are all good. Scientific journals, Nature, Science, etc. are all good. Popular magazines that are known to be reliable, Scientific American, National Geographic, Time, all good. Books and textbooks on biology, microbiology, any other textbook. Let me see if I can find a textbook. Trouble is, I'm at home. I don't have my textbook here. I don't have a textbook readily available, but all textbooks have um, a literature citation page where they have who referenced the textbook. So you can find that. So all textbooks are good. A variety of scientifically reputable television shows, like from the History Channel, PBS, are all good. But you should be suspicious and probably not use any website or document that is religiously, politically, or privately funded, because they will have their own personal viewpoint and their own personal biases. You should not use college or university pages that contain student work. If it's a professor, like you're quoting my web page, it's probably good, because if it's a mistake, eventually someone will point it out to me and then I will correct it. Okay. But if it's a student web page, the mistake gets made, that's never going to be corrected. So don't use student pages. You cannot use Wikipedia, because anyone can go into any Wikipedia page and then change it. You can use a source that Wikipedia uses. So you can use Wikipedia initially to get your project started. And then when you have to give a in-text citation, you can go look up the in-text citation on Wikipedia and then use their in-text citation. But you cannot use Wikipedia for a in-text citation or a reference, because anyone can change any page at any time. Any question about any of that? There are other online encyclopedias which are not changed, and if they are not changed, you can use them. You must give a literature citation page. This is where you have your full references, where you list all the sources cited within the in-text citation in your paper. So they should be a one-to-one match. You must give the full citations. You must use the same citation style for both the in-text citation and the full citations. You can use any citation you want, like APA, MLA, whatever. The complete literature citation page is worth 15 points. And let me show you how you do a full citation. The easiest is actually a web page, and most of you will probably give web pages, so I'm going to give that first. You give the full author's name. We're just going to say Smith, and then you can put the names or initials. I'm just going to go Smith initials, A period, oops, J period. And then if there's only one author, that's it. And then you give the date. And then the page name. Let's say something like so that's the let me go over here. And then the page address. Oh, come on. And you should have the page address there, which would be something like www.discover, I don't know what it would be. We'll go something like that. Something like that. Okay, any questions about that? So let's say this is a web page. And this is the full references. Which usually you just call it references. All right. For a journal, once again, you give the, oh, let me, let me just go use this one example again. So the name of the article, which actually that should be right here. And then the journal name, volume number of the journal, and then the page number. I'm just going to erase that now. For a book, you give the author, the initials or name, the date of the publication, the name of the article or chapter, better term, the name of the book, the editor of the book, the publisher of the book, and the city of publication. If there's more than one city, you only need to list the first. Any question about that? Name of the article or chapter, name of the book, the editor of the book, publisher of the book, and the city of publication. Now, if there's more than one author, once again, you go something like, let's just copy this again. And that would be true for all of these. You need to give the other author, if there's two, and if there's 3, 4, 5, 6, 7, you need to give all of the authors' names. There is no abbreviation in the full references. You cannot use at all in the full references. Any question about any of that? There is one exception. You may use at all in the full references if there are more than eight or more authors. That's the only exception. Okay, so there has to be 8 authors or more before you can use at all. So essentially, most of you will never use at all in the full references. Any question about any of that? All right. Like I said, I went through that in great detail because students frequently get the index citation and the full citations, the full references incorrect. You must have at least 4 unique good references in your paper. And I can generally just check to see if it's a good reference or a poor reference. But you must have at least 4 unique good sources references, or you won't get the full 15 points. So the complete literature citation page is worth 15 points. The document must be submitted in the folder on Canvas and an attachment, either a docx, MS Word, rich text format, RTF, a .write format. Any other format will not be checked for plagiarism, and that's why I'm limiting it to these documents. And don't try to submit a PDF document and then change it to docx at the end so it'll be submitted, because I'm just going to give you 0 on that if you do that. I want the document to go through a plagiarism checker, and so it has to be a docx, RTF, or a write format. Any question about any of that? A write is a format that WordPad makes. Okay. Any text editor should be able to save it down as a rich text format, an RTF format, if you cannot make a doc or docx format. Any question about any of that? All right, so the text should have context using subject subheadings so I can tell where each subheading begins and where they end. And this will be worth 60 points, this context text. If you do not have the subheadings, you'll get a -6. Okay, I really want the 6 so I know exactly where each topic begins and where each topic ends. There should be a subheading labeled causative agent where you discuss what the causative agent of the disease is. Is it a bacterium, a virus, a prion, or eukaryote? For a bacterium, you must tell me the characteristics of the cell, what the Gram reaction is, what the cell shape is, cocci, a rod, whatever, what the cell arrangement is, the growth arrangement, what some metabolic capabilities are, anything that's important. If it's a virus, you should tell me what if it's a DNA virus or an RNA virus. If it's an RNA virus, is it a positive strand or negative strand? If it's a retrovirus, you should mention whether a virus is envelope or naked, how large the virus is there a provirus, and are there any unique characteristics of the viral multiplication cycle? If it's a prion, tell me what a prion is, what the body does with this prion, what's the function of the normal type of the protein. It's a eukaryote. Is it a fungus? Is it an algae? Is it a protozoan or a platyhelminth or a nematode? Is it multicellular or unicellular? What's the life cycle? And that gives me the causative agent. The causative agent, I'll show you, it's, I think, worth 9 points. Tell me the history. How long have we known about this disease? Historically, how was the disease treated? What's the history of any vaccine development? And describe our changes in knowledge and attitudes towards the disease throughout history. Even with COVID-19, there's been a change in knowledge and attitude, and this has been a real recent disease. The attitude when it first came out was one of fear. People stayed isolated. Businesses closed down, a lot of them. People wore masks whenever you went out in public. Nowadays, it's being treated as something normal, like a common cold. Most people, when they go out, they don't wear a mask anymore. Obviously, there will be changes in our knowledge and attitude for some of the ancient diseases, as well as really any disease has gone through some changes in our knowledge. Epidemiology describe the prevalence and the transmission of the disease, where in the world the disease is prevalent, how many people are currently infected, what's the rate of new infections, what's the rate of morbidity and mortality. Morbidity is what's the illness rate, how many ill symptoms they get, mortality, how many people die from it. What's the reservoir of the disease? How is the disease transmitted? Pathology. Tell me about the pathology of the disease. Tell me about the response and treatment of the disease. Tell me about some social, political, and economic considerations. You guys can read through those. Artificial intelligence is something new. You may use artificial intelligence in this project, but you should always quote when you obtain something from artificial intelligence, even if you put it in your own words. And that's because you are quoting someone, you're quoting the artificial intelligence. And then state where you got it from. Any questions about any of that? So whenever you use artificial intelligence, you should either directly or indirectly, you must cite it. The notation that people can be characterized as left-brained or right-brained is considered to be oversimplified popular myth. This is something somebody got from AI, so they put it in quotes, and then they have an in-text citation, OpenAI 2002-3 for the in-text citation. And then for the full reference, they'll have OpenAI 2002-3, and then ChatGPT version March 14th, large language model, and then they give the reference where they got it from that web page. So the highlighted is the in-text citation, and then this would be the full references for OpenAI. Any question about any of that? I do have an instructor grading rubric, which I'll show you. I'll grade your project on this grading rubric. The cover information will be worth 2 points. The causative agent, all the causative agent information will be worth 9 points. The history will be worth 10 points. Epidemiology, 10 points. Pathology, 10 points. Response and treatment, 10 points. Social, political, and economic issues, 9 points. The quality of your writing, the grammar, the spelling, the punctuation, the flow, and the readability, if it's all good, 15 points. If there's problems with either the gramming, the spelling, the punctuation, the flow, and the readability, you'll get a penalty and it'll be less than 15 points. In-text citation, if you correctly cite information that is not common knowledge, it'll be worth 10 points. And obviously you have to have an in-text citation wherever you quote someone or wherever you quote artificial intelligence, it'll be worth 10 points. If you do not do that, you'll get less than 10 points. Like if you quote something that's not quote somebody, you state something in a fact that's not common knowledge and you don't have an in-text citation, you'll get a deduction. Like you state the number of cases in the flu in 2025 so far this year. I have no idea what that number would be. It's not common knowledge, so you must give an in-text citation. Your literature citation page, if your literature citation full references is in the correct format, you get 15. meaning you have full references. All your in-text citation has a full reference, one-to-one match. And there's at least 4 reputable sources, 15 points. If you don't have all of that, you'll get a deduction. And that'll be worth a total of 100 points. Any questions about the research project?
I had a question about the AI part.
Uh-huh.
Does it get flagged if you use AI to correct it? Like on my Microsoft 360, it has an AI to like edit or correct it. Will it show up like that? Obviously, it's not like writing it or anything like that. It would be
So
like
you
after I
wrote it.
write it.
What you're saying is you wrote it and then AI corrected it?
Like there's a button that says like kind of like spell check.
Yeah,
It it
let's
can
say you misspell a word and it corrects it. Is that what you're saying?
more in depth though, like
You're
an
quoting something and you don't get it right and then the AI corrects it? Is that what you're saying?
something like that. I'm just like
Yeah,
worried like if
that
I
is
use the
not,
AI.
that is an
Yeah.
AI correction. It's not AI generated, so you don't need to cite that.
No,
Okay.
I'm wondering if it like will like flag the paper for being like use using
Using
AI or
AI.
whatever.
I don't
Yeah.
know. I don't know. I don't know the
Oh,
answer there, so I can't answer it. But like I said, if you were to do that and you're giving it yourself, like what's an expression that people say? Oh, it's raining cats and dogs. Of course, that's so common. But let's say you don't do it correctly and you're saying it's raining cats and I don't know, poodles, and the AI corrects it, and that would be fine, meaning you don't have to cite that because AI corrected it. Okay. I don't know if AI would cite it on my end, but there is an AI checker, and if I need to, I can just submit your paper to an AI website, and then it'll show me the regions of the paper that AI generated. So it's very easy to check, is my point. So if ever you're using AI to write a portion of the paper, you should uh, put it in quotes and say you got it from AI and then cite it. And it's that easy. And that way it's loud. I'm allowing you to do it, but I just want to know that you're doing it. If you don't do it, you'll get a penalty because you are quoting someone and you didn't cite it. You are quoting AI. It's like a person. It's not, but you are quoting this AI, and so you should cite it. Otherwise, you're plagiarizing the AI. Okay? And it is very easy to check. And like I said, if I need to, I don't know if my plagiarism checker um, checks for that or not, but if I need to, I can send it to an AI site directly and have it checked independently. The AI checker may check for it. Uh, gosh, what is it? Turnitin is the AI, not AI checker, the plagiarism checker that Clark College uses. And it does have a switch for checking for AI generated text, but I can't change that switch. And I don't know if Clark College has that switch on or off. So I don't know if it will be checked. And like I said, it's very easy for me to check. I can just send it to an AI checker to have it checked. So don't, if ever you're using AI, use it, but just put it in quotes and say where you got it from, meaning put it in text and then have a full reference for the AI. All right. I think that's enough for the infectious disease. Are there any other questions? Because I need to talk about the lab. All right, let's end the index, not the index, the uh, infectious disease project. Let's talk about lab two. Lab two, lab module two, aseptic transfers. You can read about the learning objectives. I'm going to skip them because I'm short on time here. In our local environment, microorganisms are essentially ubiquitous. This is problematic when working in a microbiology lab because we generally want to work with pure cultures, a culture that contains one and only one species. Louis Pasteur developed the technique of working with aseptic transfers to minimize the risk of culture contamination. We will learn how to make aseptic transfers in this lab. During inoculation, never lay your test tube cap or your petri dish lab down on the workbench because the workbench could have microbes on it. Even if you put the cap down like this, pretending this is the cap or the lid, you put it down on the surface like that. Gosh, you can't see it. There you can sort of see it. It can pick it up on the edge. And then when you put the edge over your plate, it can fall down from the edge to the plate. To minimize contamination when inoculating media, tilt the cap. Let me see if I can show this. Just give me a minute here. I got a container I can use to work like I'm actually using something right here. So we're going to pretend this is our inoculating loop. We're going to pretend this is our tube. You can tilt the tube at an angle like this, as opposed to having it straight up like that. And then if you have it straight up, anything that comes down from the air will fall into the tube. If you have it at an angle, it would only get in if it falls directly right here. If it falls right here or right there, it's not going to get in the tube. So hold it at an angle, and that will reduce the surface area that something can fall in. And then with your plate, we mentioned this in an earlier lab. This will be difficult for me to do because I have to hold it up. Hold both the bottom of the plate and the top of the plate. You should hold it at an angle so that the top of the plate is partially covering the bottom of the plate. And then if anything falls down here, it'll go on the outside of the top of the plate, meaning falling down here, and it won't get in the plate. It would have to fall down exactly right here or exactly right here to get into the plate. So you're reducing the surface area. And then hold the lid open with your loop, which is the pencil, and then inoculate the plate like that, which they're showing you right here. Any question about any of that? That reduces the surface area, reducing the amount that the plate or tube is open, reducing the risk of contamination from airborne contamination, which if you have the lid open for just a short period of time, you're not likely to get something to fall into the plate because you need to have it open. Well, to get like 10 to 15 colonies on the plate, you actually have to have the lid open for at least 10 minutes in a normal lab. And for our lab, it's actually about 20 minutes because we purify the air to remove many of the microbes, not all of them, but many of the microbes. Sent through a filter before put in the microbiology lab. And obviously, you're not going to have the plate open for 10 or 15 minutes to get 10 or 20 colonies falling on the plate. In order to inoculate transfer cells to a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. So here's our sterile media. You only want to use a tube that's sterile, and it'll have a lid there that'll keep the tube or the dish sterile because the lid keeps the microbes from falling into the tube or the lid. And a plate, as I mentioned to some people, it doesn't ever lock or keep it airtight. The air can get in through the cracks and over, but the microbes cannot. The microbes will fall with gravity and they won't go up over the bottom portion and then over the bottom portion top and then into the plate. The air will, but the microbes will not. The microbes only fall down. And the cap, depending on how the cap is designed, some caps you can screw them on tight and have it literally closed to the air. Usually when we grow things in microbiology, we have the cap screwed loose so that air can get in, but no microbes can get in. So always use a sterile media and a stock culture that only has one species in it. And then anything you use to inoculate, like this loop here, you get it sterilized by passing the wire portion of the loop, including the loop, through the flame of a Bunsen burner, or you put the entire wire portion of the loop into the back to incinerator until the wire portion turns red hot. And then you cool the loop, put it into your stock culture, pull out your cells, and transfer the cells into your sterile media. How you normally cool the loop, the quickest way, you could let it cool for 20 or 30 seconds in the air. Most people don't do that because it actually does risk slightly contamination falling on the cool loop and then surviving. Very low risk, but it is possible to do it that way. Most people, what they do is they get their tube, and let's say the cells are on the media here. They will touch the loop to the inside of the container above where the cells are, cooling it. Usually you touch it twice or three times, cooling it by touching the side of the container and then going into the stock culture, scraping up the cells and then transferring them into the other dish. So pretend this is the stock culture. We put it in the back to incinerator until the wire portion here is red hot. Take it out, put it into the stock culture, touch it like on the side of the tube. I don't know if you can see this. Cooling it twice, put it into the cells, scrape up the cells. If it's a liquid, just grab up the loop of liquid and then put it into your other. We're going to say this is a tube, remove the cap, put it in. Any question about any of that? That's using aseptic technique. All right, know the terms of this lab. While you're not physically going to be performing any of these activities, you will be watching videos of these activities, and we will expect you to know and understand the material in this lab and in the videos. So for procedure 1, we're going to transfer sterile liquid using a glass serological pipette, shown here. And we have a little video for you to watch demonstrating proper aseptic transfer technique. So watch that video. And procedure 1, you're going to transfer 1 ml of sterile water in the pipette into a tube of sterile nutrient broth. And these are the steps here. This is a problem in the pipette, especially if it's glass, but even if it's plastic, and that is the water will not go across the pipette flatly. It'll form a meniscus like that. Let me blow this up so I can see it better. How do you read the meniscus? You do not read it from the top of the meniscus. You don't read it as the average of the meniscus. You read it from the bottom of the meniscus. So that would be 1 ml of water. We then take that 1 ml and put it into the tube of media. for procedure 1. Blow it up just a little. We'll come back to procedure 2, procedure 3 and 4. And there is a picture of the tube of the media before you put water in it. Note the color and the transparency or opaqueness of the media. Broth cultures are a problem because it is difficult to determine if there's contamination because all the cells mix together. You cannot see colonies in a liquid broth culture. Liquid culture. Okay? But if there is major contamination, you can see that the color will not be correct if the contaminant is a different color than the organism you added. Obviously, the same color, you wouldn't be able to detect at all. But if it's a different color and there's major contamination, you would be able to detect the contamination. But if there's minor contamination, you would not be able to detect that in a liquid culture called a broth culture. Now, if there's minor contamination on a solid media, you would note there would be a colony of a different characteristics than the other colonies, like a different color, for example, and you could see the minor contamination. But in a liquid culture, it's hard to see, it's impossible to see minor contamination because the color will be color from the major growth in the tube, which is what you added to the tube. All right, in tube 1, there's the procedure 1, there's the steps we're following for adding the 1 ml of sterile water to the tube of sterile media. Then we incubate the tube overnight at 30 degrees. We're going to be adding Serratia marcescens to the tube. Serratia marcescens is a good tube to use, or a good bacteria to use, because it's a bright red color. And when it's grown in a liquid broth culture, it will be a red, a pinkish color. Online students are not going to be making observations. What we'll do is show you a picture, and then you will make the observations and use this table to give your result and your conclusions. You may use the exact wording of this table, but you may use your own wording as long as you give all the information in this table, meaning for procedure 1, if the nutrient broth is clear, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. So if the broth is clear, then there are no organisms growing in the broth. You must state that, and that the aseptic transfer is successful. So you may use your own wording, but make sure you state the wording of something similar to what's in this table here. For procedure 1, if the nutrient broth is turbid, meaning hazy or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful because we transferred sterile water into sterile media. So it was unsuccessful that some contaminant got into the tube in some way or another. So aseptic transfer was unsuccessful because organisms are growing in the broth. So that's procedure 1. Use this table or wording similar to it to explain the result and the conclusion. And then this is the result of procedure 1 shown in this picture. P1 is for procedure 1. Any questions about that? And then for procedure 1, first answer question 0. Describe the initial clarity of the nutrient broth tube prior to inoculation. Was it clear or turbid? Meaning I'm telling you the answer has to be clear or turbid. And that picture is shown above, right here. And then answer question 1. Result P1. Describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees C. Was the tube clear or turbid? And then based on the results, what conclusion can be drawn? Your conclusion must use wording similar to the table above. And then that'd be 3B. So that's 3A, 3B. 3C, list 3 sources of error with a septic transfer technique that could explain why the nutrient broth showed growth of microbes. So you must answer asterisk 1, asterisk 2, asterisk 3. And the answer must make logical sense. If you say something like, something fell in the tube before I used it, the tube was covered by a lid, so nothing should have fallen into the tube until you take off the lid. Okay, so that logically cannot happen. So your answer has to make logical sense. And some of you don't always use logic in answering these questions. It has to make logical sense. Now for lab 2, let me double check to make sure this is correct. For lab 2, darn, I don't have the meetings here. For lab 2, you will have 2 attempts to get your answer correct. Let me just make sure that's the case. I think that's for lab 3 too, but let's just talk about lab 3. Yeah, I have the until date, a different date from the due date. So on the first grading of the lab, I will tell you which question is wrong. And then you have to make a correction. And then if assuming I get it graded by Tuesday morning, no, Monday morning, one of those mornings. Assuming I get it graded by Wednesday morning, you can resubmit the lab by the next Saturday, which would be May 3rd, for a second grading. So you'll get 2 attempts on lab 2. And I think lab 3 is the same. I'll discuss that next week. And that reminds me, there's a student discussion link here, which mainly this is a place for you guys to discuss different topics. A student a long time ago suggested that there be a place for students to interact with other students, and you can do this in the student discussion where you can communicate with each other. You can ask a question to other students here. You can ask a student to form a study group here. You could also email different students for that. But you can ask that. And in the first week, to encourage you to use this, I'll give you 1 extra point credit if during the first 2 weeks of the class, you post whether you want the labs to be graded 1 time as the first week labs were done, or 2 times as the second week's labs will be done. And then I'll go as the majority of the class wishes. If selecting 2 times, the lab will be due the first time, and then the first Saturday of the week of the lab, I will grade it. And after that point, meaning the lab is due that day, and that will be the due date for that lab. And then I'll grade it. And if I get it graded by Wednesday morning, and you don't have 100% on the lab, you can resubmit your answers to that lab for a second grading. And then I will grade that whenever I have time for the second grading. And then you'll get the final grade for your second grading of the lab. Usually the second grading will be done after the first grading of the labs. So the first grading will be done always first, given a priority. And so that usually means that the second grading of the lab will be held off and will not happen until about lab 9 or later. So it's usually not until the end of the term before I get the second grading of a lab graded. Any questions about any of that? But you can use the student discussion to ask another student other questions. It's especially used for students to get a study session partner. If you don't know, let me go back to my lab here.
I do
In
have a
week
quick question
2,
about the second grading.
just let me just discuss the extra credit study 2. You can get together with another student to form a study group. This can be live in person or live over Zoom or live over the phone. You may not use email. It has to be live, where you can study together in week 2 on anything that may be tested on. You may not work on an assignment together, especially the lab assignments, but you may quiz each other questions. You must quiz each other questions. And then you must turn in the date you studied, at least 2 informations about how long, what you studied, how long you studied. So tell me about what you studied, 2 sentences. And you must not study this week's lab or work on this week's lab or assignment together. Obviously, the assignment's very easy for the lecture, so you wouldn't study that. You must study something that you can be quizzed on, not this week's lab. And you must ask each other questions and then submit a document or a message in the extra credit folder for that week to get the 1 extra credit point. And then I'll go through that and read it and make sure that everyone who's in the group says they're in the group, all that, and give you an extra credit point, assuming everything is correct. Okay? If you don't quiz each other questions, you won't get full credit. If you don't ask each other questions, that's something you might be quizzed on, you won't get full credit. If you don't include the names of everybody and the complete full name of everybody in your group, you won't get full credit. And like I said, you have to tell me what you're studying and when you study it. Okay? And that can be live during Zoom. It can be live during a phone call. Or it can be live. You guys actually get together. Any question about any of that? All right. So that's the extra credit study 2. And I've mentioned that you can get an extra credit point by within the first 2 weeks answering whether you want labs week 3 through whatever lab 9 to be graded 1 time or 2 times. Somebody had a question about the second grading of the lab.
Yes, I was wondering, the second grading, does that give you full credit on it or is it partial credit?
If you get everything correct on the second grading of the lab, you will get full credit.
Okay, and that's for like the second week labs moving forward?
We are using that policy for week 2. Week 3 through the end of the term will be decided on how you guys vote. If you want the labs to be graded 1 time or the lab to be graded 2 times. It'll be on
Okay,
your
thank you.
vote. All right. So I will look at that after the end of week 2 to determine how the majority of the votes stack up. All right, that's it for there. Let me shut down extra credit study 2 and go to the lab worksheet. I think that's where we are. Oops, wrong button. Oh, Don't submit the lab module. You don't have the questions, I don't think, in the lab module anyway. Only submit the lab worksheet. The reason is the lab worksheet is much smaller than the lab module. And if everybody were to turn in the lab module, my Canvas webpage would eventually become full, in which case students could no longer submit an assignment. So only submit the lab worksheet. It's a little less bulky. And actually, I talked a little bit about the worksheet I told you, procedure 1, right there. Let's now talk about procedure 2. Procedure 2, you're going to transfer a sterile paper disc using cleaned, almost sterile forceps. Let's go back to the lab module here, because I've got it highlighted. So you use aseptic technique to transfer sterile paper disc, meaning the disc will be in a sterile plate, paper disc, been autoclaved, they're in this tube. And then what you do is you get forceps. We used to have forceps around here, but I'm not sure where they are. So I'm just going to pretend these are forceps. And what you do is you dunk them in alcohol and then burn the alcohol by either putting it in the flame or in the back to incinerator, so the flame burns the alcohol, and that very well cleans the forceps. Then use the forceps to pick up a paper sterile disc and put the sterile paper disc in the tube of media, which is sterile. Any questions about any of that? Move down a little quicker. Then use this tube to note the results for procedure 2. If the nutrient broth is clear, that means the conclusion is no organisms are growing in the broth. An aseptic transfer was successful. If, on the other hand, the nutrient broth is turbid, hazy or cloudy is the results, procedure 2 tubes, then the conclusion is organisms are growing in the broth or aseptic transfer was unsuccessful. Once again, for the conclusion, you must use wording similar to the wording in this table. For procedure 2, here's the results. The picture of P2 there. You actually see the paper disc down there. And there's the tube. And then answer the questions for procedure 2. For 2, result for procedure 2, describe the clarity of the nutrient broth after inoculating with sterile paper disc and overnight incubation at 35 degrees Celsius. Was the tube clear or turbid? And then for 2, that's 2A. For question 2B, answer the question based on the results. What conclusion can be drawn? Your conclusion must once again use wording at least similar to the table above. You may use the exact wording from the table above. And you don't have to quote it and say where you got it from. Any questions about that? For procedure 3, you're going to be transferring Serratia marcescens to a broth liquid culture. Serratia marcescens is a bright red bacteria. When it grows in a broth, it'll be pink in color. For procedures 3 and 4, we have a little video for you to watch how to correctly transfer from a bacterial stock culture to a sterile broth and a sterile agar slant, which we'll do for procedure 4. So you can watch that video there. And some notes about this video. You only need to watch the video until about 4 minutes and 23 seconds into the video. The video is using bacteria E. coli. So they're transferring E. coli. E. coli is not the same color as Serratia marcescens. It is unpigmented. Serratia marcescens is pigmented bright red. But the inoculation procedures will be the same. There is an error in the video which you should try and discover. I don't have a question there. I should make that a question in the lab module. And that is when the instructor puts his loop into picking up E. coli from the stock tube, it's between 3 minutes and 5 seconds and 3 minutes and 20 seconds. He made an error. And there's a hint you can hear sizzling when he puts the lube into the tube of E. coli. So what's the error there? For procedure 3, you're going to use a septic technique to transfer Serratia marcescens into a sterile broth culture. Serratia marcescens will grow pink in a liquid broth culture. Remember, sterile broth cultures are difficult to determine if there's minor contamination because all the cells grow together and mix together. You would not note minor contamination, but you could note major contamination. Like the tube should be pink because we're adding Serratia marcescens. And if it's some other color from major contamination, then you have major contamination. But if it's pink and there's minor contamination, you would not know minor contamination. For procedure 3, use this table for the results. If the nutrient broth is clear, then the conclusion is no organisms are growing in the broth and aseptic transfer was unsuccessful. Why is that the conclusion for procedure 3, whereas for procedure 1, if the nutrient broth is clear, then aseptic transfer was successful. For procedure 3, aseptic transfer is unsuccessful if the nutrient broth is clear. Can anyone answer that? All right, think about that because I'm not going to tell you, but you do need to know for correctly getting this question. For procedure 3, we have the tube shown here. And then answer the questions. 3, 3A, the results describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation at 30 degrees C. Was the tube clear or turbid? And then 3B, based on these results, what conclusion can be drawn? Remember, your conclusion must use wording similar to the table above. And for 3C, answer the question, how do you know whether you transferred the correct microbe, Serratia marcescens? All right, any questions about those questions? For procedure 4, we're going to transfer Serratia marcescens to an agar slant. An agar slant is a solid media. You watch the video there. You haven't done so for procedure 4. And then for procedure 4, we're transferring using a loop onto a sterile agar slant. There's the agar slant there. And you move your loop in an S-like motion along the surface of the agar here. And how you can determine minor contamination is if anywhere on the streak there's a color other than red, because Serratia marcescens on a solid culture will grow red in color. If anywhere on that streak there's a region that's not red, then you know that that region that's not red is a contaminant, because Serratia marcescens should be red. Also, if you're to find It's very hard to me to show this here. Let me blow this up as big as I can. Move this over a little bit. Oh, shoot. Move that over. Come on. I don't think I can show it here. Yeah, you're not seeing it. So I have to show with my mouse. See if I can blow this up even bigger. There it is. So if we have a contaminant right here, for example, it's not on where the slant is, then you know that's a contaminant because it's growing where there is no streak. But there's a region right here that's not red, that's a contaminant. So when growing on a solid culture, it's much easier to determine if there's contamination, even if there's minor contamination. Once again, use the table for the results. If nutrient broth is clear, use the conclusion, no organisms are growing in the broth, aseptic transfer was unsuccessful. And if nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was successful. Let me scroll down quicker. Oops, I didn't read that right. Let's go down here. If the slant does not show confluent colony growth, no organisms are growing in the slant, aseptic transfer was unsuccessful. If the slant does not show confluent colony growth, then organisms are growing on the slant and aseptic transfer was successful. There's procedure 4, the tube shown right there, and the growth is so confluent, you can't see where we even streaked. Down here, that's a little lighter color because there's a little bit of moisture there, and probably shortly after you did the streaking or during the streaking, there was water there, and that's why that's a little lighter there. Then answer the questions for 4. What result is there for P4? Is the bacteria growing on the slant? Answer yes or no. How do you know? State why you know. That's 4B. And 4C, does it appear to be bacterium inoculated or a contaminant? And then why? Any questions about this lab? If not, go ahead and work on this lab. I will be here until 8:30, although I have to use the bathroom here, but I'll be back in just a few minutes and work on the lab. After 8:30 or the last student logs out, I'll log out as well. There we go.